Hello there. I apologize in advance for my semi-pandemic face. It's a real pleasure to get to present to you at this symposium today. I'm going to be talking about work performed in my former lab when I was a postdoc at Harvard, and where we're taking this work now here at Stanford to try and build functional human-scale tissues to help children with single ventricle disease to create a Fontan biopump. A single ventricle disease is a congenital heart defect, which results in a single ventricle physiology, wherein you essentially have only one functional ventricle. And as a result of this single functioning ventricle, you have a pooling of deoxygenated and oxygenated blood. And this mixed blood ends up passing through the aorta and therefore is unable to perfuse the end organs as efficiently as necessary as a result of the low oxygen saturation. In order to solve this physiology, patients undergo a series of staged operations, resulting in what's called the Fontan circulation. Now, in the Fontan circulation, you connect the inferior vena cava and superior vena cava, which would normally drain into the right atrium. You actually connect them directly in series with the right pulmonary artery. And so now the blood, which normally passes twice through the heart in a single circulation around the body, is only passing once through the heart. And the blood now needs to travel not only through one capillary bed, through the organs of the systemic circulation, the capillaries of the systemic circulation, but also has to pass through the capillaries of the pulmonary circulation. This results in a very high central venous pressure in the inferior vena cava, and that pressure back propagates through down to the end organs. This results in venous congestion, high central venous pressure, and ultimately results in a high risk of end organ failure. Now, currently the Fontan operation is performed by inserting a passive conduit that brings blood up from the inferior vena cava up to the right pulmonary artery. This is the most common form of the procedure. There are many others, but most commonly these days, you put in a synthetic or allograft. This graft is passive, so there is no extra energy that's being supplied to the blood as it travels through the lungs. Now, our goal is to try and take this and instead of inserting a passive graft, what if we could have something that would contract? And that contraction would be able to drive unidirectional flow with a pair of valves and essentially energize the right-sided circulation, hopefully overcoming these issues of venous congestion and end organ failure. Additionally, it would be able to provide pulsatile flow to the pulmonary vasculature, hopefully reducing the gradual reduction in pulmonary vessel size that occurs in Fontan patients and lowering the pulmonary vascular resistance. Now, if we want to make something as very ambitious and sci-fi sounding as a human scale functioning second ventricle, there's enormous engineering challenges and scientific challenges we have to think about. The first is that in a typical organ, you're looking at ones to tens to hundreds of billions of cells per organ at extremely high cellular densities with many cell types per organ. If you look at just a typical histology section, most of the space of an organ is taken up by cells. They're so tightly packed, they have a volume fraction of about 80 to 90% cellular material when you're looking at bulk organs. Not only are there lots of cells, but they form structures from the tens of centimeters down to the single micron with these very complex different architectures that perform important biological functions. And the more cells you have, you may get more function. So if you have more cardiomyocytes, you can contract and create more energy for the blood. But you need more vascular perfusion to supply those tissues with sufficient oxygen. So you need a dense and pervasive vascular network to supply these tissues to keep them alive. And we can't rely on neovascularization from the host, because when we're working at centimeter scales with large human scale functional bulk tissue, it needs to be in place, ready to go. Otherwise, the tissue will die in the time it takes to implant the tissue and for neovascularization to occur over the first few weeks. So we need to be able to put the vessels in place. Now, fortunately, there have been many approaches in the last decade that are really opening up the opportunity to work on these sorts of systems. The first is iPSCs, which unlock our ability to make patient-specific cell types. The second is organoids, that we're able to now take these iPSCs and actually derive three-dimensional tissues from these that will self-assemble, form multiple cell types in the cardiac sense. There's been 3 papers recently on different cardiac organoid protocols that are generating cardiomyocytes, endocardial cells, cardiac fibroblasts, epicardial cells, and even undergo some self-assembly. And the third method that most of you probably haven't come across is this idea of embedded 3D printing. So 3D bioprinting is really changing the game in tissue engineering. It's allowing us to be able to write complex structures into soft 3-dimensional matter. And if you're printing with plastic, then you can print structures that hang in the air and are stiff enough to hold their own shape. But biology is messy, it's floppy. And so if you want to be able to work with floppy materials, you need to be able to print it not in air, but in an embedded support bath. It's like a hair gel-like material. We call this property viscoelasticity. It's a material that's not quite viscous, it's not quite a liquid, and it's not quite a solid, it's not quite elastic. It's a mixture of the two. This gel is liquid-like enough that as the material, the printer nozzle translates through it, it's not carving it like a turkey. You're not creating canyons as that material passes through. The hair gel is able to self-heal behind the printer nozzle. At the same time, it's solid-like enough that when I deposit my yellow material here, which is a silicone just for visualization, it's able to stay in place, not float, not sink, not pull in by surface tension. So it's this magical viscoelastic property of the material that's allowing us to do this really exciting method of printing where we can introduce vasculature. Now, we asked whether we could get that property not in hair gel, but if we use embryoid bodies or iPSCs, if we have them at a high enough density, do we get that kind of self-healing viscoelastic property that would allow us to directly print blood vessels into thick tissues? So working with a colleague in the Lewis lab, Sebastian Uzel, we took hundreds of thousands of these embryoid bodies. We formed them in these little microwell arrays, and we centrifuged a few 100,000 of them to form essentially a embryoid body toothpaste, a little paste of embryoid bodies, which we could extrude onto this machine called a rheometer that measures the material properties of this material. So, really you're looking at billions of stem cells here that are rendered into these embryoid bodies, compacted. And when you actually measure their property, they actually have shear thinning and viscoelastic properties, shown here by these rheological studies. So this is perfect. Now we have very high cell densities. We're working with iPSCs, and they have a material property that's ideal and conducive to 3D bioprinting. So we wanted to try and see what this looked like. What you're looking at is a bath of embryoid bodies. Each sphere is about 2,000 iPSCs, and a printer nozzle traveling through it. As the nozzle passes through, the embryoid bodies are displaced, and they re-zip up behind it. So we're not carving through the tissue, we're just moving it through, and the tissue is self-healing behind it. But if that printer is extruding gelatin, that gelatin can actually push the cells out of the way, essentially creating a path, a conduit through which we can flow oxygenated medium to keep these cells alive. So in this case, this is gelatin. So we can then raise the temperature and the gelatin remelts, and we're then able to flow liquid through that channel. Let's take a look at what this looks like, zooming out. We call this process sacrificial writing into functional tissue, or SWIFT. Sacrificial materials are ones that you lay down and later remove to create space, which is essentially what we're doing to be able to create channels into these densely cellular tissues. Now, that vessel that you see there is holding close to 1 billion iPSCs in the form of compacted embryoid bodies. And you can see that we're extruding that red gelatin ink into the material. It's neither floating nor sinking. It stays in place where we put it. So we can design using the printer any print path that we like, and then essentially remove the gelatin by raising the temperature. At the same time, when we raise the temperature, we added a little bit of collagen between the embryoid bodies. That collagen solidifies, so the embryoid bodies solidify, and the gelatin is removed, and we're able to connect it to a little pump and show here the blue dye going through the channels that we just printed. So we're able to write in perfusible networks into these tissues. We can tailor the lumen diameter so that we can make something more complex, like a vascular tree, here that we're printing inside of a silicone mold. So you can see we have a single inlet and a single outlet, and we're able to write these channels between the two sides of the tissue, and then evacuate that gel so that we can perfuse it. Now, if that densely cellular tissue didn't have any blood supply, only the outer 800 microns here would remain viable. Any further, and the oxygen just can't diffuse into the core of the tissue. However, if we have these perfusible channels, we're able to maintain the full thickness of this tissue, in this case, 4 millimeters across, in a viable state. The tissues I've shown so far are made up of iPSCs. And as you probably know, we have no iPSC-based organs. So it's important to note that this process can work with any aggregate type, so long as you can with any cell type, so long as you can make aggregates with these cells. So for iPSCs, those are called embryoid bodies. We can make a channel. You see the lumen here in cross-section. We can make cerebral organoids and then add perfusible channels into those, or even cardiac spheroids. We're going to focus today on the cardiac side of the story. So if we want to scale up cardiac differentiation, we're using this 3D differentiation process. So it's fully suspension culture. The cells are grown in agri-wells and then lifted off and then cultured in 3D and differentiated using a version of Sean Palachek's protocol. And we're able to generate little cardiac aggregates here with about 80% cardiomyocytes, and the cellular density is way up in the hundreds of millions, 200 million per milliliter of cardiomyocytes. The non-cardiomyocytes, most of them express vimentin. We think of those as sort of a stromal non-cardiomyocyte lineage. And if you look at these under the microscope, you can see these beating independently. Now, this isn't much use if I want to create a tissue that beats synchronously. So we wanted to see what would happen if we push these cardiac aggregates together. So we made a little disc out of cardiac aggregates, about 3 millimeters across. And on day one, you can see that there's very little motion occurring. There's little bits of fibrillation here and there, but not much motion. But by day 10, those aggregates have noticeably fused. And in that fusion process, they also synchronize in their contraction. So that we're able to start making larger tissues that beat more synchronously. We can look at this with calcium imaging. On day one, you have these very beautiful and chaotic patterns of signal propagation. But by day 7, these chaotic patterns are now more rhythmic. And you can see that they are more synchronized. And we can also paste these tissues, shown here at 1 Hertz. So let's take a look at what this process looks like as a complete process. So we form a tissue with a particular shape. We can now write these blood vessels into the tissue. I use the word blood vessel very loosely here. We are working on lining these with endothelial cells and smooth muscle cells. But here they're just bare channels that at least we can perfuse the oxygen and nutrients in. Let's rewind this a bit. We remove the gelatin. We show we can perfuse media through the core of that tissue. And then after 16 days of culture, it's actually able to contract very visibly. This is a centimeter tall. So you can see it kind of from the other side of the lab as it beats, and it's pulling on these little springs that we add to align the sarcomeres. You can see this vertical alignment on the sarcomeres. And we can run cardioeffective drugs through these lumens to be able to test what happens if we add a adrenergic agonist, or if we add a heptanol to block ion channels. So by doing this, we're able to test response at a larger scale. Now, with an eye on the overall prize here, it's that the 3D printing really lets us do complex geometries. So we wanted to take a patient-inspired geometry of some of the major vessels of the heart and recreate it in a wedge, as if this patient had an infarct of, say, their left main, and we could come and then write this sort of wedge-shaped cardiac tissue, but with these small vessels sort of contained within with patient-specific geometries. So here at Stanford, we're really excited to take this sort of approach and scale it to a functional level. And we're focusing on a pulsatile cardiac conduit. The design of this will need 3 billion cells and a 4 millimeter wall. And it's such a thick wall that we'll need to incorporate our vasculature through our approach. And there'll need to be a pair of valves that we 3D bioprint. we're going to be using a combination of methods derived by many collaborators here in the 3D bioprinting field, a process called Fresh by Adam Feinberg at Carnegie Mellon. It will allow us to print the valves and some of the overall shape of the conduit, and this process of Swift that lets us write in vasculature. So we're very excited to sort of take the steps towards this. So we're working on scaling the manufacture of these cardiac organoids so that we'll be able to make the 3 billion cells necessary for each practice, as well as printing some valves. And once we make the tissue, we need to be able to mechanically test it and train it. And we have these perfusion cardiac bioreactors that allow us to do this that we're working on. So this is very much our work in progress, but I hope you sort of see how the pieces of the puzzle are coming together, really enabled by stem cell biology and 3D bioprinting, and I'm happy to answer any questions. Oh, sorry, before that, very importantly to acknowledge all of my amazing collaborators at Stanford, in my prior lab in Harvard, in this additional ventures collaborative peers that we're able to work with a great group of clinicians and scientists. And I'd like to thank the funding sources of additional ventures for supporting this work. So thank you.
Thank you, Dr. Skylar-Scott. That was an amazing presentation on a really novel approach to innovate on a traditional procedure or approach to right-sided heart disease that's now decades old. And we do have a question in the chat. Can you discuss the ability to scale this amazing 3D process to human tissue size? And do you see any roadblocks with that?
Yeah, thank you. That's a great question. I think scaling is going to be a real critical challenge for our field to address in the next decade. So scaling occurs on many levels. Scaling part one is how do we get enough cells? And that is very, very expensive. Many of you scientists may be familiar with growing 1 million or 10 million cells in a dish, and then creating 1 or 10 billion cells in a bioreactor means a lot more media and a lot more expense. And then characterizing those and purifying those. If you want to purify cells using FACS, well, you can do maybe 10 million cells, but you can't do 10 billion cells through FACS. So we need some new technologies to be able to more scalably process control and purify cells of interest. And then, the next question of scaling is 3D bioprinting itself. 3D bioprinting doesn't scale very quickly when you want to make things that are large and at a high resolution. For those of you that have used a bioprinter before, or any 3D printer, you may be used to print times of 24 hours. So if it takes 24 hours to make the conduit, what happens to the cells that were laid down first, or the cells in the printer? So we need to be able to speed up our hardware. And then we need to be able to incorporate enough vasculature. To me, that's been the crux of tissue engineering for decades, is once you start getting thick tissues, how do you incorporate the fusible vessels to keep it alive? So there we'll be needing different approaches to be able to write vessels. I showed you this Swift process where we can write some large scale vessels, but how do we write the capillaries? How do we get all the endothelial cells lining in there? I think we'll have to turn to a hybrid approach, different technologies that incorporate lasers to be able to do some ablation, as well as some printing approaches, and find hybrid strategies. And then how do we test it? How do we know it's safe to put in a patient? That's going to be really, really hard. And I don't think we're going to have all the answers there. I think the whole field of cell therapies are grappling with this question, and we're excited to see how the field and how the FDA starts to consider more of these cellular therapies over time. How you can do process control and safety validation prior to implantation of such tissues. But that's it. How do we scale it? That's the 100% the right question.